Welcome everyone to the second session of this morning, which will be about machine learning methods. I hope you enjoyed the discussions during the poster session. So we directly go to our first speaker, Magnus Rattray from University of Manchester. He's a professor of computer science and machine learning, talking about spatial and temporal expression changes.
Okay, thanks for the invitation. This session's on machine learning, and this talk's going to be quite methodological. So I'm going to talk about a class of probabilistic model called a Gaussian process, which has lots of applications in spatial and temporal modeling. Trying to get this to work. There we go. Okay. So, so I'm going to kick off with a quick intro to Gaussian processes, and then I'm going to talk about applications in pseudotime inference, modeling branching, and modeling counts data and spatial data. So Gaussian processes are like probability distributions over numbers, but they're actually probability distributions over functions. And the main object of relevance in a Gaussian process is called the covariance function. And it can tell us whether the functions that we draw from this distribution are very dynamic. They could be smooth, or they could be rough. They could be stationary or non-stationary, or they could capture things like periodicity. So the covariance function controls these properties and gives us some kind of prior knowledge input into our machine learning or our statistical inference. There's a bit of a time delay here. I don't know. I keep clicking it twice and it keeps going to, okay, see if it works. Okay, so the idea is like this. So this isn't a Gaussian process. This is actually a Gaussian distribution. So what I've done is I've drawn a single vector of 25 dimensions from a Gaussian distribution, and I've used the covariance matrix on the right-hand side there. So it's a 25 by 25 matrix. And if I draw a vector and then I line it up along the indices of the vector, then what you see is that the draws from this random distribution actually look a little bit like curves. So a Gaussian process is just the extension of that to infinite. So if I take the vector of 25 and make it infinite, actually in these plots, I think I've made it 400, not infinite. And I draw functions now instead of vectors, then I see these wiggly curves. And each of those curves is a draw from this distribution. So it's a distribution over wiggly curves, basically. And the nature of the curves is determined by the parameters of the covariance function. And so we have L squared here, which is the length scale. If I make the length scale long, then I get curves which cross zero less frequently. And if I make L short, if I make that small, then I'm going to get more wiggly curves, like on the right-hand side there. So this covariance function is called the squared exponential, and it gives me these smooth curves. But I could equally have something more stochastic. So here I've got Ornstein-Uhlenbeck process, and then I get rough samples from the Gaussian process. I can put in periodicity. So here I've got smooth, perfectly period samples. So these are random samples. So if I draw another one, it's going to look different, but it's still going to be periodic. Oops. And I can also combine those. So I can take a product of the OU one, which gives me rough samples, and I can, sorry, this is going crazy. I can take a cosine one, which gives me periodicity, and I can get stochastic periodic oscillations. And actually, we use this to model oscillations in small gene regulatory networks in single-cell microscopy data. Gaussian process regression is where you combine one of these covariance functions with data, and then you can learn a nonlinear regression through the data. And as an example application here, I'm showing on the left-hand side a fit to a Gaussian process. On the right-hand side, a fit to a constant thing. Yeah, let's use this instead. Thanks. I can use a log likelihood ratio in order to get test statistics to determine whether genes are varying in time or not, just to do differential expression. OK, that's better. OK, now there's some practical considerations. So Gaussian process inference is slow if you do it naively. So we use something called sparse inference to speed things up. Sometimes you have non-Gaussian elements of the likelihood, or you have latent variables, and then you have to use something called variational inference or some other kind of approximate inference. And the methods I'm showing today are implemented in the GP flow package, which also allows you to use GPUs as well as CPUs. So the first set of methods I'm going to talk about are for dimensionality reduction and pseudotime inference. And Fabian was reminding me that he was using GPs for this in like 2012 or something like that, so a long time ago. Right, yeah. So, the twist here is that John Reed and Lawrence Vernish introduced a Gaussian process method which did pseudotime, but in the case where you have capture time information. So it's a sort of single-cell time series data set. And so they did this by having a prior on the time variable, which they're inferring along with the Gaussian process regression model. So we introduced a little variation by introducing, as well as having this capture time, also some additional covariates. So we call those X. And those can capture other variations. So you might have pseudotime, but you might also have some other variation that can capture things like branching in time. And then we did a much more efficient implementation using GP flow and these sparse variational methods, so it really scales up to big single-cell data. Now, the good thing about the GP is it really allows you to capture sort of prior information. So if you want to do pseudotime on something, for instance, that you know is periodic, then you can introduce that through the covariance function. So you can choose a periodic covariance function. So these guys took our Grand Prix package and applied it to this single-cell riboseq, and then they were using the package because it allowed them to capture this periodicity. So that's kind of a nice feature of GPs. The other nice feature is that you can use these extra dimensions to model things beyond just the time covariate. So here's some quite small scale data set, but it's good for illustration. So you have these developmental time course in early embryos. If you do a PCA, you can see that it's definitely nonlinear because all these kind of different clusters go on top of each other in 2 dimensions. Standard GPLVM just does something like t-SNE or UMAP. It just allows things to be more nicely clustered by using nonlinearity. But then if you put the informative prior on the x-axis, so you have these time labels on the x-axis, you can align the x-axis with developmental time, whereas the y-axis can capture other variation in data. And that allows you to do pseudotime along x, and then capture other variation along y. And then once you've done that, you can kind of use pseudotime to look at individual genes. And what we also noticed was that having this extra x variable actually also improves the pseudotime inference along the x-axis. So having the 2D optimization actually makes the pseudotime inference better than doing it in 1D. And the model kind of scales up because we have this scalable implementation. And then it's nice if things are reproducible. So it turned out that the people who developed Pyro, which is this probabilistic programming language, they saw our paper and they used it in their documentation for Pyro. And they actually coded everything up in just a few lines of Pyro, which was slightly upsetting because we'd spent quite a long time actually on the variational inference. But it shows it's reproducible, and they got exactly the same results. So what you saw there is when you did the pseudotime, you got this kind of branching dynamics for individual genes. And so another thing you can do with Gaussian process models is model this type of branching process. So originally we did this for just normal time series data, where you have 2 time series samples, and you're interested on discovering when those time series begin to diverge from each other. So for instance, you could have a perturbation in a time series, and you could look at when the two time series diverge because of that perturbation. So to do this with a Gaussian process, what you can do is you can create a Gaussian process where the functions are coming from 2 different draws from this random number generator or random function generator. So I have an original function in red, and then I have a new function in blue, and the blue function is constrained to touch the red function at a particular specific time, and we call that the branching time. And so this is one draw from this function generator, and then I've got another draw here, and then another draw. So I can just draw lots of branching processes that look like this from my random function generator, this Gaussian process. And so once I've done that, I can do 2 things. I can work out the probability distribution over the branching time. So I can do inference on data over where the 2 time series branch from each other. And I can also work out a Bayes factor for scoring whether something's branching over not branching. And I can integrate out all these functional degrees of freedom when I'm doing that. So it's actually quite a low-dimensional inference task. So here's an example on time series data. So I've got black dots from a replicated time series and red dots from a replicated time series. And the GP allows me to work out the posterior probability over the time where those 2 time series begin to diverge from each other. So you can see that I have this distribution over potential branching times, but it's pretty confident at some point that branching has definitely happened. Here's 2 time series that don't branch. So they are varying in time, but they're not actually branching away from each other. And in this case, the posterior probability is highest right at the end of the time point, which means basically there's no branching has occurred during the measurement time. So single-cell data is a bit different from time series data because you don't have the labels anymore for which of the time series the data came from. So you have some kind of differentiation process through pseudotime. So you need to solve the pseudotime problem. You need to work out which of the branches cells belong to. So you have differentiation, and you have to work out where the cells belong to. You want to work out which genes are involved in the branching. You know, some genes are going to be involved in differentiation. Some might just be housekeeping genes that aren't doing anything. And you want to know when things are branching. So you might have early transcription factors driving the process, and then other things changing later. So you want to kind of order the genes. And we implemented this BGP package a while ago to use Gaussian processes to do that. And the idea is that you have a branching Gaussian process. So we've got 3 branches. We've got a trunk and 2 child branches for this Gaussian process. We have an indicator variable, which tells us where the cells go. So that's a difference. In the time series, we don't have that because we know in the time series the labels of the data. We know they come from time series A or time series B. But in the single cell, we have to infer those branch labels. So we have some binary variables, and then the likelihood has to integrate over those binary variables, and that's not tractable, so we use sparse variational inference with a mean field approximation over the z's to do this. Now, in the original version, we did it independently for every gene, so it's really kind of univariate for genes. And that's a bit disappointing because it means that the cell labels can be different for different genes. So you might assign a cell to one branch in gene A and a different branch in gene B because it's a univariate model. So the model doesn't know that the cells actually should belong to the same branch for different genes. So recently, we've got a new version called multivariate BGP, which actually allows the cells to be consistently assigned to the same branch across different genes. So that Z variable there, which is the assignment of genes to branches, is shared across all the genes in the data set. And that's also computationally more efficient. So it's more correct as a model, leads to less inconsistency in assignments, leads to better assignments, and it's more computationally efficient. So it's kind of win-win. So here's an example of how that looks. It's not come out so great, but the idea is that the ground truth is in kind of magenta. I think blue is the inferred branching time. So you're inferring where the genes are branching. But you also have to infer the labels, which is the colors on the blobs. So you're kind of inferring these discrete colors, and you're also inferring the branching times. This allows you to kind of order the genes in time. So you can apply this to, if you have some differentiation process, to kind of order your genes according to where they are beginning to branch, and hopefully that gives you some kind of indication of the causal sequence of events through pseudotime. Okay, so far all these methods have just assumed a Gaussian likelihood. So we know that counts data, particularly from single cell, is sparse, it has a lot of zeros, and it is counts. Okay, so it's not coming from a continuous distribution. So here's an example of something that could go wrong if you don't take that into account. So in the bottom row here, we're just using log counts plus 1 as our measurement of expression. This is just time series data, it's not single cell. We have 2 samples, and we want to know if those 2 samples have different dynamics. Okay, so it's a 2-sample time series test. And we can do that by fitting 2 different models. In one model, the 2 time series are given different profiles, and in the other model, they're given the same profile, and then you can do a likelihood ratio test to decide whether there's dynamics or not. If you use a Gaussian likelihood and you use log counts plus 1, what happens is that that's the bottom right-hand panel here. The Gaussian has a variance parameter, and because those green points just happen to be quite close together by chance in this data, the variance is inferred quite low for across the replicates. And it means that the GP fitting the green points ends up being quite wiggly, and actually that penalizes it in terms of likelihood. So you get a lower likelihood score for the right-hand bottom panel than the left-hand bottom panel. And basically what happens is you do this test and it says the green and the blue are the same, or they're more likely to be from the same profile than different profiles. If you use a negative binomial counts likelihood, which I've done in the top row here, you have a bit more information about the variance because the negative binomial is an over-dispersed Poisson. So if you have a low number of counts, you kind of have a lower bound on the variance. So you're never going to kind of assume the variance is really, really tight on your replicates. You've got a kind of lower bound on it. And that kind of regularizes the variance estimation a little bit. And it makes things perform better. So the left-hand model, where it thinks that the green and blue come from the same profile, it ends up having much worse likelihood than the right-hand one where they're different. And in this case, the genes are clearly having different dynamics. So things work better. And there's been a bunch of papers that basically say that it's reasonable to use negative binomial on single-cell RNA-seq data, even if there is sort of zero inflation beyond negative binomial. It's quite hard to infer that extra inflation anyway. So the way we do that in a Gaussian process is that we use a link function. So in our GP counts package, we use a log link function, but Socrates here has started using a soft plus, which is a slightly different link function. And so F comes from a Gaussian process, and then you model the mean of the data as the exponential of F. So the inverse link function is the exponential of F. And you put that into your Gaussian process package, and then you have to use some variational inference because the inference is no longer exactly tractable. And what you find, so this is simulated data where you're doing a one-sample test for dynamics across time or across pseudotime, is that in the case where you have high dispersion, then using a negative binomial helps a lot. And particularly in the case where you have high dispersion and high counts, you can see that the Poisson does very badly, and the Gaussian is also not doing so well in that case. So that's kind of where you get a good win. If you have low dispersion, like bulk RNA-seq, actually transformed data using a Gaussian likelihood is not so bad in that case. So you can kind of get away with the Gaussian likelihood on bulk data, but on single cell, you definitely have high dispersion often. Now, we also benchmarked on the TradeSeq benchmark, and we're a bit disappointed. We just couldn't get good results. And then we noticed that if we used true time rather than pseudotime, we actually did really well. We sort of aced it on this benchmark. And we couldn't quite work out why. And we were trying to understand why we were getting so many false positives. And then we had a look at the data. And Slingshot's being used for pseudotime estimation. And I can't remember how many genes there are in the data, but basically what seems to be happening is that the act of doing pseudotime inference actually is inducing temporal correlations in some of the genes. So when we looked at the ones which we were calling as temporally correlated, like this one on the left here, then when we eyeballed the data, it did look temporally correlated, but it was a negative in the data set. And it kind of, it just flags up the concern that pseudotime is not time, right? It's inferred from a model, and the act of inferring pseudotime adds errors and can introduce correlations into the data in ways that you might not anticipate. So you have to be careful in over-interpreting results where you're using pseudotime as though it's time. And so, it's great that we worked well and that we got good results on the true time, but in a real application, we wouldn't have access to true time, so that's not actually going to help us too much. I think it probably depends a lot on the data set, how distributed the signal is across many genes and how many different things are going on dynamically, how big a problem this would be. I think it could be a problem in some applications and less of a problem in other applications. So you can do branching inference with the counts likelihood, and you get much more plausible, credible regions on the data. It doesn't really change the branching time inference too much. And you can also do spatial variations. So spatial DE was a package that used Gaussian processes with a Gaussian likelihood, and then we just sort of plugged in this negative binomial likelihood, and it improves the kind of detection, so you get many more detections of plausible spatially variable genes. And the ones that it excludes tend to be really low counts ones. And I think it's similar to my first example with the time series, that when you have really low counts, using a Gaussian likelihood means that you're often going to misestimate the noise, and that's going to lead to problems. Whereas because the negative binomial sort of has the Poisson as a lower bound on the noise, it can deal with the low counts cases really well. And we can scale this up. So this is some Visium data using sparse variational inference. And now we're really interested in sort of extending that to multi-output Gaussian processes where we really need it to be scalable, and that can kind of capture, clusters and multivariate signal in the data. But that's ongoing work that Socratia here, wherever she is working on. So you can talk to her about that if you're interested. So to summarize, Gaussian processes are useful for modeling things like branching, periodicity, and spatial effects. Basically, if you want to do machine learning, but you have strong priors over spatial and temporal things that are going on in your data, they're a good choice of model. The uncertainty allows you to do things with a level of confidence. So if you're temporally ordering events across pseudotime, then you can do that with these uncertainty levels. And if you have the ability to model the data well using appropriate likelihoods, then that's going to improve performance. In terms of challenges, in the branching across pseudotime, we inferred pseudotime first and then did branching, and it would have been nice to do them together. That'd be really nice. You can take gradient observations. So in the GPLVM, for instance, if you have gradients from something like RNA velocity, you can actually include those in the GPLVM model. It'd be really nice to, we've implemented that, but if anyone has a good application, we'd be interested in talking about that. And you can also use things called latent force models, which take into account differential equation modeling within the data. Again, RNA velocity might be a good example of that. As I said, multi-output GP models to capture correlations across genes. And I think scalability can always improve. Some of the methods, admittedly, are a bit slow, and I think that probably limits the uptake among practitioners. So we still have to work on that. And just finally, thanks to the group members and collaborators and funders, and thanks to all of you.
Thanks for a great talk. Questions? Fabian?
Thanks, Magnus. This is pretty cool. I've been, so I think particularly also the multi-output part for the spatial setting, I think sort of picking up regions there. People have been coming up with some of these approaches. I think Ollie has one with the spatially early on, right? Wonder a little bit about that. But my first question was on these gradient observations. So we always had the impression that if we maybe don't make the best choice of K, that this interpolation sometimes tends to wobble quite a bit, and you saw some of these oscillations happening in between. So if you really use the gradients from the GPs, I guess the uncertainty could be quite high. So you think we could use them for an ODE type of model, such as for these transcription rates and so on?
Yeah, so I don't mean using the gradient of the GP. I mean using gradient observations in the data. So if you have a data point and you have its location and its gradient, or any derivatives, you can include them in the GP inference. And that would actually improve the wiggly that would improve
regularize, right?
That wiggliness problem. The other thing is that wiggliness, I think, comes from the use of the RBF covariance a lot. And in the past, we've used other covariances like an integrated process that's non-stationary. We did that in some time series for that very problem. So we had a time series, and our covariance was an integrated RBF, and it tended to not have such wiggly behavior between the data points. So yeah, I think you can design covariances to not have that wiggly behavior.
Yeah, the last thing I wanted to say is, like, if you have real time series information in addition, right, now that you have a real functional description of the gene dynamics, I guess that would be also straightforward, or at least possible to include, right?
Yeah. Any kind of prior information is easy to include. Yeah, definitely.
Interesting.
Yeah.
Beyond the OT stuff that's usually happening at the moment, right? So
Yeah.
a lot of stuff is happening with optimal transport. I think if you have sort of a generative model like this one, you could be more descriptive.
Yeah, I think so, yeah.
I have also a question I can ask. So it is not very intuitive to me if the Gaussian process can be used to explain non-monotonous gene changes. For example, if you have a treatment, you have a response, and you go back to. So can we use Gaussian process for these things, or do I always need a monotonous component instead?
No, actually, they're ideal for the case of non-monotonic changes. So a lot of people have got time series model where they've basically hard-coded certain behaviors in, like sigmoidal or like something that goes up and down, that type of thing. But the GP can capture all those functions anyway. So it can go up and down and in fact, what's harder in a way is constraining those things. So it's actually harder to make a GP that is monotonic. And you can do it, people have done that, but that's a little bit more challenging. So if you know the data should be monotonic, the problem is the GP might come back down again, So that's, yeah.
Can have one more, Maren, maybe?
Yes, beautiful talk, thank you. I was wondering, can you, if I have, let's say, two, parallel processes like cell cycle plus proliferation and differentiation, we have quite some problems with disentangling those. So can you actually do that with a GP?
I think you should be able to. I think the problem might come if they interact, which they probably do. So for instance, people have shown processes which tend to happen. At the beginning, I showed this oscillation, which is actually this HES1 gene in neural progenitors. And I believe there's been some work that shows that the oscillations often kick off after a cell cycle or something. There's some interaction. So if they're not interacting, I think it's quite easy because they're just different covariates which you can model, which are kind of orthogonal. But if they have a relationship with each other, it might require a little bit more sophistication to do it. But I think you can definitely do it. I'd be really interested, if people have good problems like that, I'm really interested in collaborating on that. I think that's really interesting.
Okay, thank you very much. Our next speaker is Florian Büttner. He's a professor at the Stanford University and talking about interpretable machine learning.
All right, thank you very much for the introduction, and also thanks a lot for the invitation. I'm very glad to be here, even though I haven't been working on single-cell data all that much recently. So in fact, we're interested really in the application-driven development of machine learning algorithms, and today I'll really focus on the algorithm part. And specifically, we're interested in uncertainty quantification, calibration, and domain shift, how these things interplay, and how we can get a trustworthy and interpretable model. So in the first part, I'm really going to focus on these method-C things, but I do promise I'll show some single-cell data in the second part, so bear with me. But first of all, what does it actually mean when you talk about trustworthy and reliable machine learning models? And I'd just like to give you a couple of examples what I mean when I mention these words. So what you can see here is the image of a skin lesion. Well, on the top left, it should be pretty obvious. And it's fairly straightforward to train a classifier that has a very good accuracy in terms of distinguishing this lesion as a benign lesion from a cancerous lesion. And so if you get a prediction with a confidence score of 99.8%, you think, okay, cool, yeah, that works. I can trust that, right? But actually, what our collaboration partners did, they just zoomed into that image and then gave it to the same algorithm. And then it said, hey, well, actually, I think it's cancer, and I'm 93% certain. And unfortunately, this model was wrong about 50% of the cases when you did zoom in. So I think we can all agree we can't really trust that model, right? Because if we were able to trust it, then it should be able to tell us that it's kind of uncertain. And if it's uncertain in about half of the cases, then the confidence score should be around these 53%. We wouldn't mind that it makes a wrong prediction because it has never seen these images before, but you want to know that it's not quite certain. So this is another example to illustrate that a bit further, and just a little toy MNIST in this case, where you have like a pretty perfect accuracy when you train your neural network, and if you give it some image from the test data set, it'll make a prediction with a pretty high confidence, which matches the very high accuracy. However, if you now take this image and sort of distort it a little bit, what you'll find is that at the beginning, on the top left, you can still see that the confidence scores are very high. The model makes right predictions, matches accuracy, all well and good. However, once you get to the right-hand side and you distort the image that it's not recognizable as a digit anymore, the model suddenly makes wrong predictions, which is fine. It has never seen it before. But it makes these wrong predictions with a very high confidence, and at the end says, no, I'm actually 100% certain this is a 2. And this is, of course, problematic, because very often in practice, when we deploy a model, we do see some kind of domain drifts, and we never know how they're gonna work out. And the same thing is something we saw also for the melanoma data. So when we trained a model on data from Barcelona, applied it to data from Sydney, we found that we had the same problem with making these wrong predictions with a very high confidence. So we came up with an approach that allows our model to actually communicate uncertainties. So basically what happens is that at the beginning you still make these confident predictions and it's all good, it matches accuracy. And at the end you still make wrong predictions. We can't avoid that, the model has never seen it before, but it's really able to tell us now that it's actually not that certain that the last image is a 2, because it sort of has to make up that there is like this missing part, and right. So before I tell you how we managed to train that kind of model, I'd like to talk very briefly about how we can actually quantify the quality of the predictive uncertainty of our models. And we usually do that with the concept of calibration. So we can compute the calibration error, which tells us how well a model is calibrated. Calibrated just means that the confidence or the confidence of a model should match the true likelihood of a prediction for all levels of confidence. And I can quantify this with or visualize that, first of all, with a reliability diagram shown on the left, where basically I take all the confidence scores and I put them in bins. So the second to last bin contains the confidence scores between 80 and 90%. And if a model was calibrated, then this bin, all the predictions in that bin, should have an accuracy between 80 and 90%. However, in this case, just the ResNet trained on CIFAR 100, we have an accuracy of only 40%. So the model systematically makes overconfident predictions. And this is data from like just a seminal paper from Kilian Weinberger's lab from 2017, actually. And what you can do is you can then summarize these reliability diagrams by building the weighted sum of these gaps. And this is basically the ECE. OK, so how did we manage to get our model to make uncertainty-aware predictions with a low calibration error? Well, we said, well, our model doesn't know how to be calibrated, so let's teach it and alter the loss function. And so to this end, we included 2 new terms in the loss function when training the model. First, we encouraged a uniform distribution of the scores in case the model does not know, so just like maximize the entropy. And then second, we explicitly encouraged calibration for all types of out-of-domain samples. And to this end, we generated sort of generic out-of-domain or domain drift samples using adversarials, very simple adversarials, and importantly, only optimized the ECE rather than the reconstruction error. We evaluated calibration across different types of dataset shifts, including completely out-of-distribution data, and found that overall our model really performed very well, covering the entire spectrum of gradual domain shift from small shifts to truly out-of-distribution data. And we had sort of an improvement in ECE over Bayesian neural networks, as well as deep ensembles of, on average, 30%. And this is just like an example where you can see sort of like a number of other baselines that are trained already to have a good predictive uncertainty, but still make very overconfident predictions under domain drift. Okay, so that's kind of useful, but the problem is you really have to train your model from scratch, right? Because you have like these new terms in the loss function. So very often what you do have is an already trained model, and the question is, can I somehow sort of post-process the predictions of this model to make them more reliable? And there exists the concept of post-hoc uncertainty calibration, which has been around for quite a long time. You may be familiar with plot scaling in the context of SVMs, for example. And the idea is basically to take, an SVM is a distance to a hyperplane, or for neural networks, you take the logits, and you somehow transfer them, for example, by dividing them by a temperature. And then these transformed logits should be more calibrated. And yeah, as I said, temperature scaling, we just divide the logits by a number is the most popular way of doing this, and this works like very well. But the problem is, even if you do this, so you fit this temperature of the validation set, and then you get more calibrated predictions, this only works really well for the in-domain case. For the distribution shift, you still get like very high overconfidence. So we came up with a way where we, again, generated a perturbed validation set mimicking a generic domain drift scenario, and fit our standard post-hoc calibrators, and then got a 50% improvement in terms of calibration error. So this is just an example of how it looks like. Here we have like an image from ImageNet, where we have like 1,000 different classes from sort of everyday objects, like a doormat, as you can see. But please stay here. And if you look at the confidence of an uncalibrated neural network, you get 99% certainty that this is in fact a doormat. And if you use baseline calibration, same thing, our proposed method as well. But now, if you use an image taken from the ObjectNet database, which basically has the same classes, but sort of taken a different angle and different context, so stuff you experience in real life deployment of these models, then the model is suddenly uncertain and makes wrong prediction, which is fair enough. This, it could be a handkerchief, but the uncalibrated version is very certain again about it, with 91% confidence scores. If you do this standard temperature scaling, you still get a very confident prediction of 85%, and only with our approach, you have like some kind of notion of uncertainty that okay, maybe you have to be careful here. And this also works on the average. We applied the whole thing to the melanoma data as well, and this also worked quite well in a similar manner. Right. So what I've tried to show you now fairly briefly is that there are some ways where we can train uncertainty-aware deep neural networks which are able to reliably communicate their uncertainty, both under domain drift scenarios when you have some kind of perturbations and leave your training domain, but also for in-domain cases, of course, and also how to post-process already trained neural network to make sure that their uncertainty, their predictive uncertainty, is actually reliable under all cases. And yeah, so they're really well calibrated for any degree of domain shift from these in-domains to truly out-of-domain samples. We haven't really thought about much about applications in single-cell data, to be honest, but I saw some interesting posters on uncertainty in cell type classification, and yesterday also some people talked a bit about uncertainty in regression problems. So if you have any ideas, I'm happy to chat about applications. Right, but before I move on to the single-cell part of the talk, I'd just like to give you maybe a quick caveat. So I've talked about how we can use this ECE to quantify calibration, and maybe we thought, okay, use a histogram as a metric, isn't that a bit? And we systematically now looked at how well actually these different types of calibration errors that are out there do quantify calibration and the quality of predictive uncertainty. And turns out, not very well, because there's a number of ways to do that, including the ECE that's shown in orange, and all kinds of other methods as well. And so what we did here in this paper was to say, okay, we train a neural network, we fix the model, we train a post-hoc calibrator on a validation set, we fix it, and we recalibrate it. And everything's fixed, predictions are fixed. And now we want to quantify the calibration improvement on a test set. And all we do is we vary the size of the test set. So once we compute the calibration improvement on 100 samples, and then up to 10,000 samples. And what you would hope is that, because nothing has changed, the calibration improvement is the same. This is just the number of samples we use to estimate the improvement changes. You would hope that it's stable. But actually, all these errors are really biased and highly depend on the dataset size. And you can see that the red line, which is an upper bound that we derived in this work, is the only unbiased way. And so you can really see, the more samples you have, the more you overestimate your calibration improvement. And for the ECE, we also have some theoretical work on why that is. And so the way we came to this upper bound that's actually stable and non-biased is that we said, okay, well, clearly all these calibration errors have nothing to do with proper scores, but actually we can relate each calibration to a proper score. And once we do that, we are also able to derive a quite tight upper bound. And if we use that upper bound, it's provably unbiased, and we can get these nice straight lines. We also get to use these calibration errors for optimizing recalibration and then get better recalibrated scores, and we can use it in the regression case, which I also quite like. So I'm going to very briefly show that. So this is just a UCI data set regression, like the housing data set, I think, where you predict housing prices, I think. And basically, what we did is a mixture density model. So we have a predictive mean, but we also have a predictive variance. And what we did is we monitored the predictive variance over the training. So at the beginning, the model doesn't know anything, and you would hope for a high predictive variance, right? But when you train your model, well, you have like quite a flat variance. That's the blue line in the left plot there. We then used our framework to derive a calibration error based on the DSS score, which basically generalizes the MSE. And you can see that the recalibrated model actually is able to capture the model uncertainty with a very high predictive variance at the beginning, which then gets lower along training, where our model gets actually more certain and really captures the behavior it's supposed to do. So yeah, I quite like that work. And if that's interesting to you, come and talk to me. Right, okay, so I'm really going to switch gears now and talk about something that I've been thinking about for many years and been interested in many years, and that is how we can use latent variable models as discovery tools. So latent variable models basically reduce some kind of high-dimensional data set of correlated variables into a low-dimensional data set of uncorrelated, but hopefully interpretable latent variables. And I think this interpretability is key, because very often what we do is, we may fix the variation, we may fit the variation autoencoder and get our 30-dimensional embedding, then put it UMAP on top of it, have this 2D space, and then you look at some clusters that we see in the UMAP. But it's actually very hard to interpret what has been going on in the VIE, what the latent space actually means. And so you can use all kinds of post-hoc explainability stuff, but I believe another way that's also very useful potentially is to design models to be inherently interpretable. So I want a model where I infer a latent factor that I can look at and directly interpret rather than having to go via some clusters and some UMAP. So, well, a good way to come up with interpretable factors is a linear model. And in the easiest case, you take a matrix, you decompose it as a weighted sum of some factors. So, well, in our case here, we don't have patients, we have cells times genes, and we have a weight matrix and some factors times samples matrix. This is a very, very simple model. And what we did next is we extended that to the multi-view case. So in the multi-view case, you don't only have like one matrix, you have like several matrices. So you have like your for example, single cells, and you measure the transcriptome, and you also do a TAC-seq, so you have multi-ohm data, so you have like two matrices, you share the number of cells, have different features. And then we can decompose it into weighted sum of our factor matrix, where the factors are shared between all the views, and you have like weights that are specific to a view. And so if you only look at the top line, this is basically the MOFA model, in case you're familiar with it. And what we did is to say, okay, one problem with MOFA is that you really can have a hard time interpreting the factors, because, okay, you can look at the loadings and you can do some gene set emission analysis, but you can get big lists then. So why not like design the model that we use already, the prior knowledge that we have so vastly, as we heard yesterday already. And so we said, okay, we just take gene sets or pathways from like, say, rectome, MCTQE, KEK. We encode them as sort of a binary matrix where we say, okay, if a gene is annotated, we set it to 1. If it's not annotated, we set it to 0. But we also know that these pathways are noisy, and we don't know how noisy they are. So we better be Bayesian about it, because we don't want to get garbage out if you put garbage in, right? So we say, okay, we just use sort of a sparse prior, and the model can decide if the pathway is correct. It can refine the pathway. It can switch off genes that are not active. It can switch on genes that are active. And then once we fit the model, then we can use it to visualize the factors, which then actually correspond directly to pathways. And we can look at the factors, and we can refine the factors. So it's quite a nice tool, I think. We did that by modeling this structure sparsity via a modified horseshoe prior. We designed it to use for it to run, well, primarily on patient data, so low sample sizes, but it scales very nicely to hundreds of thousands of samples because we use stochastic variation inference, and there's some nice theoretical properties. So first of all, we compared it to other sparse models, right? Because this modified horseshoe prior is not the only way to induce sparsity in a model. And in particular, MOFA has a spike and slab prior, BAS has a 3-parameter beta prior. Then you can use like a VAE where you put a L1 regularization, you also get some sparsity, so there's lots of ways to do that. So we then looked, okay, how well do these models actually recover sort of the true sparsity pattern that we can simulate, and how well are they able to reconstruct the data? And what you can see here in this image is that basically, our model has the sweet spot in terms of having a very high F1 score in terms of recovering true sparsity, but also having a low reconstruction error. So the BAS model actually also works quite well in terms of F1 score, so it recovers the sparsity very well, but it's not so great in terms of its reconstruction error. And then MOFA and the autoencoder have a good reconstruction error, but they're not working very well in terms of recovering the sparsity. And what's really important is for movie, we have 2 crosses here, right? For 1 cross, we didn't inform the model at all, so we have like an uninformed prior. And still we get quite a high F1 score, like a bit better than BAS actually. And then the other one, we give in pathways, but with a 50% noise. So we give wrong pathways that corrupt with a 50% ratio. And even the super corrupted pathways still allow us to improve the model. So here's some analysis on the TCGA data set. So again, not single cell, but next slide, I promise. And what you can see here is that, well, first of all, we can do like a t-SNE on the latent space that we infer, just to make sure that our model really reconstructs the data well and captures the global structure, namely that we have the separation of cancer types, which it does. And what I forgot to mention is that what we did, we ran it by annotating only the mRNA view in this multi-view data with MSigDB and KEGG, because we want to see what happens if you also have like an unannotated view. And okay, so much more interesting is of course like the factor view, where you can directly look at the factor space, and 2 very important factors were here, antigen response and melanogenesis, that explain a lot of variance, because we have a nice ARD prior that explains us, or tells us which factors are most relevant. And what you can see is that, right, so there's lots of gray dots as well that you can't see, unfortunately. But what you can see is that, well, antigen response really separates nicely the prostate cancer cases from all the rest. And then in the y-axis, there are 2 different types of skin cancer in the TCGA data, namely UVM, SKCM, and these are also very nicely separated. So I'm afraid you have to believe me or look at the paper. So what you can do is then actually go a bit deeper and have really this local view and say, okay, now you tell me antigen response, but which genes are driving it? And I can tell you, well, so these markers, and actually the model refines this gene set by switching on more cancer-specific genes as well, and really refines that pathway for our application. We can do the same thing. Oh no, sorry. And as I said, we only inferred the mRNA view. So the nice thing about this model now is that we can sort of transfer the pathways to a different view. So we can now see how does actually the antigen response pathway look in the proteomic view. And then we can see, okay, well, these genes are being switched on, and they make a lot of sense. These are a lot of genes related to antigen response in the context of prostate cancer. So we really have like this transferred pathway to different views. And of course, this also works with all kinds of other views if we have them. All right, now finally towards single cells. So we briefly ran our model on 10K PBMC from profile with a 10x multi-ohm kit, and annotated the RNA-seq view with the hallmark gene set and reactome. And so again, we ran a UMAP on the latent space. Okay, the model really nicely captures the global structure again, so we have a good reconstruction error. And more interestingly, we want to know which pathways are driving the variability. And then for the RNA view, you get lots of like immune system stuff. And for the ATAC-seq view, unsurprisingly as well. But you can also see there are some differences. For example, vincyclilin is only active in the ATAC-seq view and not in the RNA view. And then we can still again do our multi-scale analysis where we look into, like, in this case, the most commonly or pathways explaining the most variants, and you can see, okay, allograft rejection, just really this strong immune response, separates activated T cells and natural killer cells, makes a lot of sense. But we can also, or we have a functionality in movie where you can give in sort of either cell clusters or covariates, and it finds the pathways that are explaining variants and differentiate between these two classes. And we did this for two types of dendritic cells, myelodendritic cells, MDCs and PDCs. And we find that MHC class 2 separates really the MDCs from the PDCs, and this is like well known that HLA is activated in MDCs only. And similarly, neutrophil gene granulation separates the PDCs, where again we have like this interplay between neutrophils and dendritic cells. And we can also look at the weights that are driving this, because maybe for neutrophil degranulation you say, oh, really? But for example, the top gene that's annotated here is known to be a marker and is specifically lowly expressed in PDCs and highly in MDCs, which is exactly what we see in this factor. And then for MHC class 2, we see that we have lots of HLA being active, and the model again switches on some related genes that are not part of the pathway, but working in a coordinated manner. So we can really, really nicely refine these gene sets as well. We can also learn sort of de novo gene sets by having unannotated factors and dense factors to capture like confounding factors, which I'm not showing here, but this also works quite nicely. Right, so in the last 2 minutes, I rushed a bit because I really wanted to talk about this after Magnus's talk, because I always have some GPLVM stuff. And so I don't have to go into detail to it because you very nicely introduced that. So the thing about GPLVMs is that you basically model the covariance between the samples, but you don't really explicitly model the covariance between the genes. And clearly we know that there is a covariance structure between the genes. So the question is, can we also make that explicit? And so we use the GPLVM, which I don't have to say that is a sort of GP regression with an unobserved input, on which you use the kernel then to get your predictions and reconstruct your gene expression. So what we did now is we said, okay, well, we can also introduce a kernel to model the covariance between the genes. And so what we do, we don't only infer latent variables for the samples, but also latent variables for the genes. And we have a new kernel that works on that to identify like the covariance structure between the genes. Now, if you do that, and people have thought about that before, is you have to connect these kernels via a Kronecker product, which means it scales horribly, and you couldn't ever use it for single-cell data. But we actually came up with a nice way of coupling the inducing points when we do a variational inference, so we have like very nice scaling properties of this model, actually. We published it last year and used it to predict movie ratings, but we have since extended the model for the multi-view case and applied it to single-cell data, and also for the imputation of proteogenomic data, where it also kind of works nicely. So this is just like to place it in the context of the GPs. So our model basically combines the GPLVM with like the LVMO-GP, where you have sort of now also dependency between the outputs via a latent variable, and you do your co-regionalization by kernel. So very last slide, done a second. Basically, we ran it again on the PPMC data, and you can see that, now we have like only 2 latent variables that we infer, and we can directly look into it, and we can see actually if you have now this nonlinear model, it's enough to have 2 dimensions to separate all of these cell types without doing a UMAP. And the advantage is that you can still use all the GPLVM stuff that Florian and I did many years ago, like gene relevance map, and make them really nicely interpretable and know what's going on in the latent space. But what we can also do is we have now our gene embedding, so we can sort of link the clusters that we see in the cell embedding to the gene embedding. And what you can see on the right-hand side, again, unfortunately, there's lots of gray points that are missing, is that if I look at differentially expressed genes for the dendritic cells, they all cluster together here in in the gene embedding space. But actually, there are also lots of other genes inside that circle that are unfortunately not visible, and that allow you to directly sort of complete the network, because we know now from this joint inference that they also are tightly connected with each other. So it's like basically a complementary way of looking at the gene space and the cell space simultaneously. Yeah, and with that, I'd just like to summarize quickly that these latent variable models can be used to infer unobservable hidden states in an interpretable manner if we take this prior knowledge into account. Please, if it's interesting, check out our movie package. Aba has done a great job in making lots of tutorials. It plays nicely with muon as well, and integrates in the NData and ScanPy world too, so check it out. Finally, thanks to Christian, who was an intern with me at Siemens back in the day and started the uncertainty work, Aba, Sebastian, and Sarah from my group, and Yinchong from Siemens AI, with whom I worked on the GPU stuff. And if you want to join, we have a postdoc and PhD position available as well. And happy to take questions if there's time.
Cool, thanks, Florian. A very nice presentation. Also, I think very important for the single cell community to try to integrate reliability and interpretability into our models, right? So if you don't know the answer, tell me I don't know. Don't give me random, right?
That's the idea, yeah.
I think that's very important. And then the last part about the GPL. Are there, yeah, Magnus?
All right, that's really nice. And what's your feeling now with the GPLVM? Yeah, clearly the drawback before was that each variable was its own GP function, which is too over-parameterized.
Yeah.
So you're capturing these correlations. Does that make it more sort of competitive with VAEs now? Because it's sort of able to really do high-dimensional data properly?
Yeah, I think it does. But so I think it's different ways about thinking about dimensionality reduction, because when we use the VAE, okay, we infer this 30-dimensional latent space, but no one ever looks at that latent space, right? Everyone just does a UMAP and looks at the UMAP. And I think the GPLVM is a way out of that we can actually go back to the latent space, directly look at the latent space, because I think it's much more, or it can be more meaningful and interpretable. So, and if we can do that in a competitive manner in terms of, the clustering performance and whatnot to the VAE, then I think it's a good approach. And I think this is where we getting competitive and have the big advantage of having directly the latent variables.
Just one more question.
Yeah.
Do you think it improves local optima? Because actually in my talk where I showed the PBMC that we did with the GPLVM, it was the 2D GPLVM space. It wasn't a UMAP.
Yeah.
But we initialized it with t-SNE, I think.
Oh, did you?
And we ended up with a better representation in t-SNE. But if we just started from scratch, I think we got into bad local optima. So do you think your method also is better for local optima?
Possibly. We never initialized with something nonlinear. Either we did random or PCA.
Because I think one problem with GPLVM is because you've got so many functions to learn, you can get stuck in a local optima really quickly.
Yeah. No, I remember from the old days where we ran vanilla GPLVM. Yeah.
Okay. One very short question, maybe more. Okay.
Hey, thank you for the great talk. In your first, one of the first models you showed, Falcon, you need adversarial examples. So with images, that's clear. I was wondering how you would think about that in a single cell context.
So we actually designed it it's modality agnostic. We also use it in text data, for example, because we use the simplest type of adversarials. Our aim is not to be robust to attacks, right? So we just say, we compute the gradient with respect to the input, and then we just remember the sign of the gradient, and that's it. Put a bit of noise in the direction of the gradient. So on images, we put Gaussian noise on it, but works for tabular data as well.
Okay, really cool. I'd be excited to try it then.
Yeah, let me know how it goes.
Okay, cool. Thanks. Let's move to the short talks now, and then at 1, we will go for lunch. Actually, we will take 5 more minutes. We started 5 minutes later, so yeah. And our speaker is Yusuf Roohani from Stanford, and he will talk about multi-gene perturbations.
Thank you. Thank you for attending the talk, and I want to thank the organizers for giving me this opportunity to speak here. It's a real honor. Thank you. So my name is Yusuf Roohani. I'm a PhD student, and I'm excited to talk to you about our work trying to predict novel multi-gene perturbation outcomes using transcriptional data. This is joint work with Kashin Huang and Yuri Leskovich. So one of the leading textbooks in the field of molecular biology begins with the words, we understand the structure of the universe better than the workings of living cells. We still cannot predict how a cell will behave if we mutate a previously unstudied gene. And this fundamental problem in molecular biology is very closely related to what we're trying to solve in this project, which is what is the transcriptional response of perturbing a combination of genes that you've not seen experimentally perturbed before. And this problem also has a number of biomedical applications, especially in the design of patient-specific therapeutics. So for instance, in the field of drug discovery, it would be very valuable if you have knowledge of multi-gene perturbation outcomes to be able to design perturbations that could reverse a disease phenotype. You could also identify genetic interactions. So for instance, synthetic lethality is the interaction where if you perturb just one gene or if you perturb two genes in combination, you could potentially kill a cancer cell. But if you perturb each of those genes individually, you would probably not get the same effect. Another area is regenerative medicine, where re-engineering of cells has become very useful in the design of cell-based therapeutics and also in potentially reversing phenotypes linked to aging. But one of the key challenges that are preventing experimental approaches from really answering these questions is the combinatorial explosion in perturbational space. So if you just consider all pairwise combinations of known protein coding genes, you're already looking at 4 × 10 to the 8 possible combinations. And if we scale this up to higher orders, we see that this goes up by orders of magnitude. So clearly, to predict the vast majority of combinatorial perturbation outcomes, we need computational approaches. So the first step for us was to formulate this problem mathematically. So we asked, what is a genetic perturbation and how are we formulating it? So consider some unperturbed cell with gene expression values G1 through Gn, shown here on the left. And what we then do is apply a genetic perturbation to some subset of these genes, shown here in red. So in this case, you're perturbing G2 and G4. And this perturbation could be either an activation or a repression of those genes. And so our goal really is to be able to predict the outcome of this genetic perturbation. So it's simply a function that takes as input the initial state and then the applied perturbation, and it predicts for you the effect that's happening across all the other genes. Now, this problem of trying to understand the transcriptional relationships between genes is not a new one. There have been so many talks today and yesterday that have tried to look at this question from many different angles. One angle that has been particularly popular has been this idea of learning the intrinsic structure between genes and their transcriptional relationships. So the popular gene regulatory network inference algorithms try to do this, and one approach to predicting an outcome of a perturbation is to infer this network first and then kind of try to predict using that network what will happen if you perturb some of the genes in that network. But the problem there is that it's very hard to learn these networks for large perturbational data sets. So there's been a paper recently that showed that the performance for these algorithms on single cell data sets is actually not very good. The other issue with these models is that they're not able to capture complex nonlinear genetic interaction relationships. There's also been some recent work that looks at deep learning-based approaches that will try to use a latent space to skip the network inference step entirely, and they'll try to predict the outcomes directly. But these approaches also have their own shortcomings in the sense that they don't have a meaningful representation of the perturbation. So they'll often rely on one-hot encodings and therefore will be entirely dependent on the training data to make sense of the perturbations that they're seeing. So if they see a new gene perturbed that they've never seen perturbed in their training data, they don't have any sense of what does that mean. So we present a new approach, which we call Graph-Enhanced Gene Activation Repression Simulator, or GARES for short, which leverages the best of both of these approaches. So we're a deep learning-based model that uses large single-cell perturbational data sets to learn how to predict gene perturbation outcomes, but at the same time, we are constrained by prior knowledge of gene-gene relationships. And what this allows us to do is to be the first computational method that's able to reliably predict perturbation outcomes for genes that were never perturbed in your training set. So in the next 5 slides, I'll go over some key aspects of our model and how they use biological intuition to build inductive biases into our machine learning approach that go on to then improve our performance in a number of different tasks. Okay, so the inputs to our model are, as I mentioned earlier, the unperturbed gene expression and the genetic perturbation being applied. So the first step that we do is we create multidimensional embeddings for each gene and each perturbation. So most existing single-cell-based analysis approaches will usually just use a scalar representation for each gene. But what we found is that there's a lot of heterogeneity in the response of different genes to perturbation. And so we found that it was actually more meaningful to represent each gene and each perturbation with a multidimensional embedding. This gives the model more capacity to capture gene-specific heterogeneity, and also is very helpful for detecting genetic interactions. So then to simulate a perturbation, we'll simply take the gene embeddings, and we'll add to those gene embeddings the specific perturbation embeddings that are being applied. So in this case, gene 2 and gene 4 are being perturbed, so you simply add the embedding for gene 2 and gene 4, which is P2 and P4, to all the genes in green, so which is G1 through Gn. And this is simulating your perturbation, and you can see that this is generalizable to arbitrarily large perturbation sets. Now, a key innovation in our approach is that we are able to leverage prior knowledge of gene-gene relationships. So the way that we do that is that we use the gene co-expression graph for incorporating prior knowledge for the gene embeddings, and we use the gene ontology graph for incorporating prior knowledge for the gene ontology embeddings. And this is functionalized using a graph convolutional network. And so each node in these graphs is a gene. And the node embeddings in these graphs are either the gene embeddings for the gene co-expression graph, or their perturbation embeddings for the gene ontology graph. And really, the intuition that we're trying to build on here, and this was also came up in a few talks yesterday, is that we need to think about genes as sets and not as like individual genes. And you can see this in like perturbation data sets, right? So this, if you create a heat map of perturbations versus genes, you'll see that there are many genes that produce a similar effect upon perturbation, and there are many genes that respond similarly to a perturbation. And so we're trying to incorporate this intuition into our model, where the gene co-expression graph is enforcing that if you have a similar expression pattern to other genes, then you're likely to respond similarly. And the gene ontology graph is enforcing that if you're involved in a lot of the same pathways, you're likely to produce a similar effect. Right, after this step, we incorporate what we call a cross-gene layer. So up until this point, we've only been working at the gene by gene level. And so we wanted to make sure that the genes have an opportunity to look at what's happening to the other genes as well, so across the whole transcriptome. So we call this the cross-gene layer, and it allows you to capture secondary effects and to generalize them across perturbations. And finally, we have a gene-specific decoder, again, to capture gene-specific heterogeneity. We didn't want to share the decoder across all the genes, and this tended to work much better. So this is our end-to-end pipeline. For training gears, we use a simple mean squared error loss. So you see on the far right there, we're basically looking at the error between our prediction and the true expression value. And the true expression value is determined from the data set. So we're mainly using perturb-seq data sets, which are basically large pooled screens of CRISPR perturbations. And so each cell undergoes either an activation or a repression of one or two genes, and you look at the full transcriptome readout after perturbations. So that's the data set we're using. And we essentially are averaging this loss across all the perturbation cells and genes. And then we also have a term in the exponential that allows us to focus on differentially expressed genes. Because the problem becomes a little simpler if you're only focusing on all genes, where most of the genes don't do anything. So you tend to instead focus on genes that are more differentially expressed. And this also helps our model in predictions. The other thing to keep in mind is that a mean square error loss has no sense of directionality. You could be predicting all the directions wrong, but still have zero error. So we have a separate term to incorporate directionality losses to make sure that we're in the right direction. OK, and this is the overall result. This is the overall architecture. Now we have some time for results. So consider we're perturbing this combination, FOSB and CEBPB. So what this plot shows you is the change in gene expression over control. So the green dotted line is the unperturbed control state. The black boxes correspond to the predicted post-perturbation expression for this particular perturbation. And then the red dots correspond to what Gears is predicting for this particular perturbation. And these genes are the top 20 differentially expressed genes for this perturbation. And what you see is that across the board, we're capturing the right trend, both in cases of upregulation and downregulation of genes. Another interesting thing to note here is that CEBPB was not seen at the time of training. So this is a completely new scenario, which has not been done in the past, where you've never seen this gene perturbed before, but you can still make a prediction for what would happen if you perturb it in combination with something else. So we wanted to formalize this setting a little bit more of not seeing some genes at the time of training. So what we did was we created this formalization of the different generalization categories. So in the bottom, what you see is if you're perturbing two genes, X and Y in combination, there are three possible settings in the training set, which you see in the top row. So it's possible that you've seen both X and Y perturbed individually at the time of training. It's possible that you've only seen one of them perturbed individually at the time of training, or you've seen none of them. And so you can see how going from left to right, you're actually increasing the difficulty of generalization. And what we find is that our model is able to show an improvement of over 45% over all baselines across these generalization categories. So we look at two different baselines. One of them is a linear co-expression graph. So essentially, we just parameterize the co-expression graph to try and predict the outcome of a perturbation. Another model is a deep learning-based network, but it doesn't use prior knowledge. Okay, the other thing that I was actually more interested in validating was, are we able to actually detect nonlinear interaction effects? So this means, like, if you're perturbing two genes in combination, a naive model would simply say, oh, the effect of perturbing these two genes is simply to add their effects, and that's your combination. So for instance, in this case, if you're perturbing PTPN12 and ZBTB25, they're showed by these two hatched bars, you would expect that the combination is just the stacking up of those two bars, right? But the reality is, if you look at the true gene expression, it's actually much higher. So this is a synergistic effect that you see in the real data set. And what we find is that GERS is able to detect this synergistic effect. So you can see that GERS doesn't make a naive prediction, it actually detects the synergy. So we looked across 20 most unexpected interactions, and we found that GERS was able to pick them up quite well. In the first three cases, you see synergy. In the fourth case with HISTH1C, you see a suppressive effect that's also being picked up. If you go right to the end, the second from the right is TUFM, and that's a neomorphic effect where essentially the single genes were not showing any effect, but the combination does, and even in that case, GERS is able to pick that up. So we also generalize this to a bigger setting.
Maybe you have to wrap up. Like.
Yeah, I think I have 3 minutes.
No, I mean, like maybe 1 more minute. Can you wrap up?
Okay, sure. Yeah. So we also looked across all the genes. So for instance, in this setting, we're looking at one gene at a time. So here we're looking at all genes together, and so now we can treat every perturbation as a vector. And then using vector math, you can basically determine whether the resultant of adding two combinations is more or less than what you would expect, and you can detect an interaction. And across all these settings, also we show a significantly improved performance in detecting these interactions. And lastly, just I wanted to share like what our vision for this tool is that we want to make it a tool that can multiply the information that we get from perturbational screens and also help experimentalists better design these screens so that they can get the most informative points experimentally tested. And so here's a pipeline that they can use. Come to my poster for checking out how we actually applied this, and we found new phenotypes that actually were not even present in the training set, but were able to be detected by our algorithm. So I'll skip all this. It's on the poster. So in summary, we present the first computational approach to reliably predict perturbation outcomes for combinations of genes that have not been experimentally perturbed. And we uncover a substantially larger region of combinatorial perturbation space, and we help in the design of new perturbation experiments. Here's the link to our code, and I think the preprint link is somewhere in the README. And I'd like to thank my advisors and my collaborators, and thank you for listening.
Questions, Malte, maybe?
Thanks, that was a really, really interesting talk. I'm wondering, to generalize this to unseen perturbations, the moment you're using kind of gene perturbations, you're using the gene ontology graph. This obviously doesn't cover all genes and has missing, potentially kind of erroneous associations. How do you deal with that? And have you looked at how that works for things that maybe are not as well represented?
Yeah, so that's a good point. In our paper, we have this analysis that shows that when the lesser connectivity you have in the graph, the more likely you are to have a poor prediction. So I think the way we caution users is to try to make predictions for genes that are more well connected. The other thing we do is we have an uncertainty loss, so we have a Bayesian formulation, so you can switch on the Bayesian formulation and your prediction will then come with an uncertainty attached to it. So you can also use that to get a sense of how much you want to trust this. But like the graphs that we use, they're actually just a suggestion. So we've done experiments with other graphs as well, and you can flip these out based on what you're looking for.
Thanks.
Yeah.
Fabian?
Yeah, thanks for a nice presentation. Thanks for building upon these sort of single perturbation models that we've been doing. You might have missed one. We did one for drug combinations where we also use graphs as a prior. It might be fun maybe to compare sort of how the prior was formulated there. This was obviously for drugs, not for genes, but would love to see your feedback on that.
Yeah, thanks for the suggestion. We've so far only focused on genes.
I think totally makes sense.
But yeah, it would be interesting to see how it stretches to drugs. Yeah.
Okay, good. Let's move directly to the next speaker, Sebastian. University of Tübingen, talking about t-SNE and UMAP.
Thank you, Lale. So actually, I'm still at Heidelberg University. I will move to Tübingen next year. And I would like to talk to you about our work on unifying t-SNE and UMAP. And this is joint work with Jan-Niklas Böhm, with Philipp Behrens, Dimitri Kobak, and Fred Hamprecht. So in many single-cell data analysis, what you start with is something like a count matrix. You all know this. And since many, many genes or other features have been measured, this is a high-dimensional data set. And now in, I would say, 2/3 to 3/4 of today's, or like all the talks you've heard, an integral step is to visualize such a high-dimensional data set in low dimension. And there are two prominent candidates. You can do a t-SNE plot. Alternatively, you can do a UMAP plot. And both show interesting structure, but over the last few years, it has been a bit unclear as to what exactly their relationship is. Also, what might be their relative strengths and weaknesses. And in our work, we show that both of them can be seen as just 2 points on a whole spectrum of embedding methods, UMAP using more attraction, t-SNE using more repulsion, and you can actually interpolate from a UMAP to a t-SNE plot and extrapolate from in both directions as well. All right, so the overview of my talk is that we will start with UMAP's true loss function. Then we will discuss, or equipped with this loss function, we will discuss the relationship of UMAP to t-SNE, and then come to the spectrum of embedding methods, neighbor embedding spectrum. So first, a bit of background. What are these neighbor embeddings? Neighbor embeddings is a framework for visualization methods, and they share many steps. You always start with your high-dimensional data. In my case, high-dimensional is 3 on this slide. And then you build a notion of similarity in this input space. I call this P here on the slide, and this is proportional to the edge weights in the k-nearest neighbor graph, hence the name neighbor embedding. And this is the information that you extract from high dimension and that you use to inform your low dimensional embedding. To get this embedding, you initialize in some way, and then you build a counterpart, an embedding similarity distribution. I call this Q here, and that depends on the distance between embedding points, such that low distance, as you would expect, is high similarity, and high distance is low similarity. Now you've got these two notions of similarity, and the main part of all of these methods and the main reason why they differ is the way you match these two distributions to each other, so the loss function. You want to match Q in some way to P, and since Q depends on the embedding positions, that means you shift around your embeddings, and in the end, you get your nice UMAP or t-SNE plot. And while there are some choices involved in steps B, C, and D, I would strongly argue that the main part, that the main difference is in the loss function. This is where the meat is. So let's check out UMAP's loss function. This is what we did. And to our surprise, we found that the implementation of UMAP actually implements a different loss function than the one presented in the paper. And this is an illustration not on single-cell data, but again on the MNIST data set. So this is images of handwritten digits. And if you take your favorite UMAP implementation and you run it on MNIST, you get something like on the right, nice 10 clusters for the 10 digits, all well and good. But if you implemented the purported loss function from the paper, you get something like in the middle. a plot devoid of any structure, really. And the reason is this discrepancy in loss functions. And we worked out the true loss function, so we went through the implementation and checked out what is the loss that they implement. And we find it has drastically less repulsion. And this reduced repulsion allows UMAP to actually find these clusters and not just have a diverged embedding like in the middle. So it's good that UMAP has this much reduced repulsion in its implementation. There are a few consequences of this reduced repulsion, a phenomenon we call overcontraction. And here again, illustrated in a very toy example. Unfortunately, you can't quite see this so well. So we had samples from a uniform ring in 2 dimensions, so a 2-dimensional data set. And then we applied UMAP to it, again, getting a 2-dimensional data set. And what you can see somewhat is that the embedding is not uniform anymore. It's kind of clumped together. And the reason for that is the diminished repulsion makes any similarity appear maximal. And for the maximal similarity, you want to have embedding points to coincide in position exactly. And that's why these clumps form. And also the ring width is reduced. And what's important from a practical side is that this phenomenon gets stronger for larger data sets. So now we have 5 times the same ring, again in 2D, and then embedded into 2D with UMAP. And we see now a perfect circle as a result. So stronger overcontraction than for the single ring. And I think this is noteworthy because it's common practice to apply UMAP on a subset of your data set. And according to this analysis, this changes the strength of the overcontraction. Nevertheless, we now have our true UMAP loss function in place, and we can try to relate it to the t-SNE loss. And the best way of doing this is by checking out how exactly this embedding similarity distribution Q is defined. t-SNE does the straightforward thing. It has these similarity values, and then it divides them by the partition function. This gives you a nice normalized probability distribution. The downside is that you have to sum over n squared many terms. And this is, of course, prohibitively slow for the large single-cell data sets you are working with. Therefore, some approximations are necessary. And one of them is NCVis. And NCVis uses a slightly different loss function than t-SNE. And the upshot of that is that you get by with a learned normalization parameter. So instead of a sum over n squared many terms, you have a single parameter that you learn. And some theory by Gutmann and Hyvärinen establishes that you still approximate t-SNE. So this is now much faster in terms of complexity. And well, equipped with this knowledge, this is all prior work, we thought, how does our true UMAP loss function fits in? And we realized that UMAP actually does the same thing as NCVis, but with a twist. And that is that we don't learn the normalization parameter, but rather we have a fixed normalization constant, and that is huge. And in terms of effect, and the size of the parameter constant is huge. And if you want to have a probability distribution, Q, and you have something in the denominator that is huge, you also need to have a large numerator. So you need to really move your embedding points close together to get high similarity to counteract the large normalization constant. This is another way of explaining why UMAP has so much attraction. And now, summing up this slide, UMAP has more attraction than NCVis because its normalization constant is much larger. NCVis approximates t-SNE, so all in all, we see UMAP is like t-SNE with more attraction. So we were quite happy with this analysis, finally getting t-SNE and UMAP on common ground, understanding their relation. But there was one thing that bugged us, and that is the apparent choice of a parameter in UMAP. So why choose this particular Z bar? Could you get away with using a different one? And turns out you can, and the result is that you get a whole spectrum of embedding methods. For higher Z bar, you get more attraction. For lower Z bar, you get more repulsion. And we see that these embeddings have qualitatively different properties. If you use more attraction, if you're more to the right, what you see is the more continuous global structure of your data set. This is a developmental data set, and in the more attractive plots on the right, maybe panel D, you see the time trajectory quite prominent in your embedding. So this is great, we see some continuous global structure. The flip side is that locally such an embedding looks really squished together. There's not much structure within these islands of cells. And that is, again, the phenomenon of overcontraction. You can counteract this by cranking up the repulsion. And if you look at, for instance, panel B, you now have much more resolved local structure, much finer local structure. It's like zooming in locally. The downside is that we lost the nice global time direction. So this spectrum implements a trade-off between continuous global structure and more local, maybe discrete structure. And for the corresponding values, or for the proper values of Z bar, we can actually nicely match our UMAP plots, our t-SNE plots, and our NCVIS plots onto the spectrum. We can get intermediate values, we can interpolate between the two. And I think it's important to say that a large range of this spectrum produces meaningful embeddings, not just where UMAP sits, not just where t-SNE sits, but also between and a bit in both directions. If you go to extreme values, you lose an insightful embedding. So if too much attraction just gives you points, too much repulsion blows everything up and you have no structure. But there are many, many useful locations on the spectrum. Okay, so this is it from me. Let's sum up. We saw that first, UMAP's loss is actually different than stated in the paper, which is good, but it also leads to overcontraction. With this true loss, we could establish that UMAP is like t-SNE, but with higher attraction. This higher attraction, if you like modulate the attraction and increase the relative size to repulsion, you get a whole spectrum of embedding methods. And the spectrum implements a trade-off between the more continuous global and the more discrete local structure. And I know this is an uncomfortable lesson, but I would really urge you to inspect multiple points in this spectrum, because if you explore a data set, you never know what features it really has. And looking at different points in the spectrum might bring out different things, might show you that some stuff that maybe the UMAP plot showed was more artifactual because it's gone in a more repulsive plot. Okay, I would like to thank again my collaborators, Niklas Böhm, Philipp Behrens, Dimitri Kobak, and Fred Hambrecht. And I thank you for your attention. I think a great place for lunch is poster 16 over there, if you want to dig deeper into all of this. And I'm happy to answer some questions now.
Thanks, Sebastian. Very interesting. Do I understand it correctly? Because if I remember, like the cost for UMAP is different, as you say, to take care also long range distances, right? So you talk here about like how the distribution the low dimensional distribution function is made, and which is different. And so you say what they do here destroys that loss function, because for t-SNE it was just a KL divergence. Now they symmetrize it for 2. So you say this thing cancels also what the design of the loss function.
Yeah, unfortunately, at least a large part. So if you read the UMAP paper, it appears that a big part of the contribution is a very careful definition of the weighing of the k-nearest neighbor graph. And so here we did some histograms. In blue on the left panel, you see the distribution of these carefully weighed k-nearest neighbor graph. And in green, you see the distribution that our theory predicts, all squished to value 1, again, over contraction. And in orange, you see that. And in green, you see what the embedding does. And yeah, it's not matching up with
And this is because they wanted to make it faster.
Yeah, exactly,
Okay.
yeah. And both because they wanted to make it faster and I mean, I don't know what their thought process was, but accidentally, they also made a loss function that works, because the original one, remember this blown up plot in the very beginning, while it would reproduce the similarities, it's just not a good embedding. It just has way too much repulsion.
Cool, thanks. One more question, the last one. Yes?
So you mentioned it would be nice to test different contraction parameters. I was wondering, there was a paper out a couple of years ago that where they start with different repulsion parameters, first having the ones that capture global structure and then going to the ones that capture more local structure or vice versa. Have you compared what kind of results these multiple parameter optimization in different steps leads to, if it's somewhere in the spectrum, or is it something quite different?
So I think I can maybe say two things on that. Is that perhaps the PackMap paper that you're referring to?
I don't remember, but they basically try to optimize in first steps with, I think, lower or higher contraction, and then the next steps with the other one.
So when I was not quite honest with you, in all of the plots at the top, and with all of the plots here, really, I use something like early exaggeration. So I use more attraction for the first third of the embedding, and then only for the last 2/3, I use the actual value that the spectrum would dictate, because otherwise I get very fragmented clusters for the high repulsion part. For the high attraction part, everything is fine. For the high repulsion part, you need that. It's like you also have to do in t-SNE. So this is kind of a hierarchical scheme here, but just with a single step, essentially. And that's necessary, yes.
Cool, thanks. Let's thank all the speakers. Very interesting session.